what I'm going to be doing is I'm going to be providing a slight introduction to spatial transcriptomics, the motivations behind performing it, and some case studies for applying spatial transcriptomics, before kind of setting the stage for Evan and Stephen to really talk about the technologies. I won't go into too much detail about how these technologies work, but I just wanted to give everyone an introduction. And so I think a lot of people come at this question of spatial transcriptomics from single cell, and it's really motivated by the fact that single cell transcriptomics has been revolutionary for our understanding of cell types and states, because it gets us this comprehensive molecular look at the cell types and programs that the cells are running within them, and it allows us to connect basically kind of our anatomical, morphological, phenotypic understanding of cell types with molecular information about what genes define those cell types. But I think all of us who really in biology, and especially those of us who grew up looking down a microscope in our training, know that tissues are not dissociated single cells. They have structure. The relationships between cells really inform their function and their state. And tissues are also very dynamic, right? Cells can move, they can migrate and divide. And so when we apply single cell technologies, however powerful they are, we kind of lose both the spatial and temporal context that these cells live in. And so what's the goal of spatial transcriptomics? Well, it is in some sense to take these molecular understandings of cell types, which a lot of us abstract in these t-SNE plots or UMAP plots, and really bring us back to the world of this abstraction is really describing some tissue architecture and how the cells are organized together. And for a long time, and however we end up taking this complex issue and dissociating it, and I think for a long time we've described tissues through the lens of microscopy. And so, Ramon y Cajal 100 years ago looked at the retina and said, hey, here are different cells which have different morphological features, and we might look at them and how they're connected, and that might inform us about how the retina functions. And for a long time, maybe the next 100 years, we've built up microscopy tools and molecular labeling approaches that allow us to look at a handful of genes at a time and generate kind of mappings between like some molecules and find spatial architecture. But as I said, maybe the goal is to connect these 20,000, there are 20,000 genes in the transcriptome, right? And using those 20,000 genes, we can create cell type annotations and generate these abstract UMAPs. Like, is there some way to bridge these two worlds, right? Like, this is imaging 4 genes. Can we image 20,000 genes? And so that's the ultimate goal of spatial transcriptomic approaches. And so today I'll tell you a little bit about why and how, and basically, how and why we do that. And so, so spatial transcriptomics, measuring the transcriptome cells without the loss of that spatial context. And there's 2 general approaches currently in the world of spatial transcriptomics. There are imaging-based approaches where you use microscopes and sequential imaging to reconstruct the localization of RNA. And there are sequencing-based approaches wherein you use barcoded arrays or barcoded pixels and to reconstruct the tissue architecture through high throughput sequencing. But at the end of the day, both of these provide spatial gene expression profiles. What that means is that for you might imagine your tissue has an XY component, and at each XY location, you get some sampling of the genes that are expressed. And so today's session, actually, Evan will be talking about giving an overview of the NGS-based approaches, and Stephen will be giving a an overview of the imaging-based approaches. But I'll just briefly say that in general, the spatial capture transcriptomics or NGS-based approaches, the concept is that basically you can encode pixels which capture RNA. So it's a little bit like turning a camera and like a microscope or a camera. Instead of taking pictures of photons, it's taking pictures of molecules. And generally how that's been done is barcoding XY pixels and then capturing the RNA and associating the barcodes. And Evan will talk to you more generally about how these methods work and how one might use them. On the other side, in imaging-based transcriptomics, it's really reliant on, at least for RNA, many methods rely on hybridization of fluorescent probes, really pioneered by Arjun Raj and other folks on how to image single mRNA molecules using fluorescent probes. I think the way to convert kind of this microscope into something that can image a couple of genes to something that's like a sequencer can image many genes is to do the microscopy many times and then register those images together. And Stephen will basically tell you about the different approaches and how we can really encode hundreds to thousands of genes with kind of this combinatorial microscopy. So this kind of sets the stage for the technologies. I'll briefly talk about now, and also maybe just to take a step back and put these technologies in context in terms of spatial scale. You can think of single cell omics as kind of like losing all spatial context, while spatial capture transcriptomics really is happening at the lag scale of millimeters to microns, although some newer technology are pushing towards maybe sub-micron resolution. While imaging-based transcriptomics really is at the scale of nanometers to microns, wherein you can capture subcellular organization of mRNA, as well as how they organize within cells and then bringing it all together into tissues. I think we all think of these as complementary approaches in terms of there's some trade-offs in throughput and capture that Evan and Stephen will explain. But in general, imaging-based transcriptomics are generally higher resolution with a little lower throughput, and spatial capture transcriptomics are generally at the cellular resolution, but with higher throughput. Now, I think, for the rest of the next kind of couple of minutes here, 10 minutes here, I'll just tell you about kind of what are some questions that you, as users of spatial transcriptomics, might tackle. And I think there's 3 broad classes of questions, and then framed in the context of biological perturbations. And the 3 questions are cellular composition, basically what are the cell types and in what are there spatial organizations or distributions within tissues. And I think many folks who are performing spatial transcriptomics are thinking about this in the context of tissue atlases of healthy and diseased tissues, especially in the context of these single cell atlas efforts. The next is cellular interactions. It's this idea that maybe a cell's state is influenced by the cell's neighbors. And that's very important, obviously, in development, in niches of differentiation, and definitely in for example, in cancer. And so that's to understand cell-cell interactions and kind of what is a syntax for describing the organization of cells and how they change in disease. And lastly, I think an area of special interest for many is cell-cell signaling. Like, because we have this prior on the localization of cells, we might then be able to answer questions about communication between nearby cells in terms of gene expression, and as well as receptor ligand interactions. I think this is an area that really merits a lot of future technology development. And so these types of 3 levels of organizational questions can be framed in the context of case control experiments in biology. What we really like to know is how are cells and tissues organized in healthy context, and then be able to ask how they change in perturbations such as disease or aging or drug perturbations, and then come up with a framework to test each of these things, right? Like how does a cellular composition change, like changes in cell type composition or where they're distributed? How does like neighborhoods change, and how do cellular interactions change? I think this is like the big picture computational and analysis goals of after collecting this data. And I think just I want to highlight a couple of case examples where people have been doing this. There was a beautiful study that came out a couple of weeks ago in Cell with a new spatial capture transcriptomic method called Stereoseq. And there they really piloted and looked at cellular organization throughout development in mice. And they looked at and assigned cell types and cellular compositions to many organs throughout this process of development. And this was done actually also in combination with a single cell or with leveraging existing single cell atlases, right? To now assign where those cell types live and maybe also then go and discover, for example, new organizations in the brain or recapitulate kind of existing known gene expression profiles in cellular organizations for example, in the spinal cord and kidney. So here, this is a question of like using spatial transcriptomics to assay the cellular composition and gene expression profiles within a tissue. And then, here we're also doing it over many time points. So there's this question. Discovering cell type gene expression patterns and cell type localizations. And then there's this question of how do you do this cell type assignment to spatial locations? I think this is a problem that many people have realized is a zeroth order problem to solve after collecting spatial transcriptomic data. We've come out with some methods for robust cell type decomposition. This is just, at the end of the day, it's just a model that allows you to ask for any given spot pixel, whether that's an imaging pixel or a spatial transcriptomic pixel, which Evan and Stephen will describe in a second, how you take those and explain what the best proportion of cell types actually explain the data at each given pixel, given a single cell reference. And this is how we can take the spatial transcriptomic data and register it to our single cell atlases of cell types, as well as then deconvolve out when the spatial pixels capture more than one cell type. And there's been many methods actually developed for cell type assignments to spatial locations. In fact, because this is such an important problem, and there was a recent paper that came out a week ago or two that's really benchmarked many of these methods in a variety of simulated and existing data sets, which will pass on to you in the notes. And I think the takeaway is that many of these methods perform quite well, including RCTD and cell-to-loc and spatial DWLS. And probably this is a problem that's important, but also there's lots of good tools for it. And the other thing I want to say is that most of these in this review paper, when you do look it over, is that there's many algorithms for cell type assignment. And the other thing is that, there's many methods for collecting data, but it turns out that most methods are actually robust to all types of data. So, you can use, RCTD, and this is applied RCTD to a star map. This is an imaging transcriptomic data set, even though it's developed for a spatial capture data set. And you can use methods that are developed for spatial capture for imaging and vice versa. And many types of these methods are actually quite robust to the data type. And I think the next thing I think that's important to note is that after you have these cell type assignments, I said we are interested in cell-cell interactions and also receptor ligand changes. And I think the next problem is that I think we have to understand spatial differential expression changes within cell types. Because we have these tools now to map cell types. I think an important question is, how does the gene expression within a given cell type change as a function of its location in the tissue? There's a lot of variability in gene expression, for example, in single cells, and how much of that variance can be explained by their interactions with their neighbors or their spatial proximity. Here, I'm actually showing you some fish images. This is single molecule fish images of a gene, ALDOC, which is known to be patterned in Purkinje cells. So this is a spatially differential gene in Purkinje neurons. And in Bergman glia, actually, this gene is still also expressed, but it's non-spatially differential. And so in single cell, what you would see is that there's a lot of variance in Purkinje expression of this gene, but that variance can be explained by spatial localization. And in Bergman, you would not be able, the variance is not explained by basically the spatial localization. And in general, we want to come out with like approaches to discover that. For example, how does gene expression change as a proximity for one cell type to another? I'm just hiding like this as one type of problem that one could do with spatial differential expression. Here, Dylan in my lab actually ran an analysis in the spatial transcriptomic data set of tumor cells. And he looked at basically how does tumor cell gene expression profiles change as a function of their proximity to monocytes, a myeloid cell type. And he found that basically that there's, upregulation of genes that are in the epithelial to mesenchymal transition, especially when those cells are next to monocytes. And you can confirm that with histology. And in general, you can imagine that you can take many types of spatial differential expression problems or questions about how cells are organized and reframe them into spatial differential expression problems as covariates. For example, you can imagine having discrete regions in your tissue or some continuous gradients or cellular microenvironments, which are explained by relative proportions of cell types. And I showed you cell-cell interaction, but also maybe proximity to pathological structures like plaques or injury. And I think so this is an area where you can convert biological hypotheses to spatial differential gene expression patterns. And so I think just to sum up here, I think the big vision is that, in the past decade, there's been many, many genomic measurements for everything going from single cell for transcriptomics to epigenomics to cell nuclear architecture. I think we can move all of these into the realm of tissues through these spatial genomic assays. And I think that's an exciting future to look forward to. And just to contextualize that a little bit and even bring together a little bit of the talk, I think we're very interested in contextualizing spatial genomic measurements by measuring a variety of different things, including tissue atlases, subcellular localization, but also genomic context, which we'll hear a talk from Julia later, and then also single dynamics of gene expression, which we'll hear a talk later as well. And so that's just framing today's section, and I'll take it away. I'll let my speakers take it away with the actual content.
Well, thanks, Faye. In the interest of time, I think we'll proceed to Evan and save questions for the panel discussion afterwards. So I'm excited to introduce Evan Macosko, an institute member of the Broad Institute of MIT and Harvard, where he directs a lab in the Stanley Center for Psychiatric Research. Evan is also an assistant professor and attending psychiatrist at the Massachusetts General Hospital. As a postdoc in Stephen McCarroll's lab, Evan developed DropSeq, a single-cell transcriptomic method. His lab now invents and applies genomic technologies, including the in situ transcriptomic method, SlideSeq. With his perspectives for both neuroscience and technology development in single-cell and spatial contexts, I'm eager to hear his talk today about spatial capture approaches. So take it away, Evan.
Thank you, Sophia. Thank you, Faye, also for introducing some of these concepts, so it'll be easier for me to get into some of the details. So I'll be talking for a little bit now about, in detail, about these spatial technologies that utilize next generation sequencing as a readout for gene expression. And Faye already covered this kind of duality that we are currently living in the sort of spatial genomics world, where we either apply the microscope directly to the tissue, as is sort of seen in the variety of in situ hybridization or in situ sequencing-based strategies that have emerged, or we kind of take the microscope out and put it into the use the Illumina sequencer essentially as the detector, and rely on the connections amongst barcodes as a way of detecting the locations of individual transcripts. So we call those sort of sequencing-based or SST technologies. And the first one of them, I've borrowed this image from the 2016 Stahl et al. Paper from Joachim Lundberg's lab that utilized a microarray strategy, a barcoded microarray strategy to capture RNA locally at 200 micron pixels. The advantage of this particular family of technologies and why it has become so appealing to many of us is that it offers whole transcriptome coverage. So by utilizing a oligo-DT probe or a combination of targeted probes, you can grab the entire transcriptome, all RNAs, onto primers that have a barcode on them, and then affix that primer on with reverse transcription. Another really key aspect of these technologies is their ease of performance. They're incredibly well suited to a lab that has a sort of like basic molecular biology and biochemistry experience, because essentially the slide is sort of pre-indexed. All of the work has been done up front. I'll talk about that in a minute. And then the downstream user can sort of treat this slide with the appropriate kind of reagents and buffers and the like, as if it were just a standard molecular biology reaction and standard library prep. And so this really takes a lot of the complexity and instrumentation out of these assays and makes it really deployable in a lot of different contexts in biology. Relatedly, another really big advantage of these techniques is its throughput. So because the imaging is again done up front and it can be done serially in very high throughput, once a slide is made, it can be, in the case of SlideSeq, which I'll talk about, the technology we developed with Faye, the slide can basically be put in a tube, and an individual operator can kind of handle and process these libraries as they would an individual molecular biology reaction. So they can be massively multiplexed to, dozens of these slides in a single day's work. And another sort of appealing aspect of these technologies is their potential to be extendable to other modalities of data. And we've already started to see this, and I'll cover it in some detail in some subsequent slides, that the combination of barcoded antibodies, for example, allows you to capture not just RNAs, but those barcodes directly onto the slide so you can detect the locations of individual proteins. Relatedly, you can use TN5 to barcode and tag individual parts of the DNA and then sequence DNA at the same time or instead of the RNA. So the ability to detect biomolecules beyond just transcriptomics obviously makes it potentially very appealing as a platform. Another appealing thing, especially for those of us who study tissues like the human brain that have very high amounts of autofluorescence, is that by taking the microscope out of these reactions, you don't have to worry about imaging artifacts. So the challenges of photo bleaching or having to photo bleach autofluorescent components of the tissue is no longer a problem. And we see that as particularly advantageous, especially for human brain and other tissues that have these problems. And then finally, the sort of like main consideration and the thing that we really want to be cognizant of is the question of resolution and diffusion, because we're capturing RNA as it emerges out of the tissue. We either actively sort of release the RNA through the application of proteases, or we use a freeze-thaw process to kind of grab the RNA out. The RNA is coming out of the cells. And so it has the ability to move not just directly down at the sort of orthogonal point of closest contact with the slide, but it could also diffuse in the XY directions. And that could create a fuzzy image or an imperfect representation of RNA locations. And it also creates important challenges for reconstructing the individual transcriptomes of individual cells. So that's something we just have to be aware of as we do our analysis, and also as we think about the differences amongst different technologies. So there have been a sort of a bevy of different individual technologies that utilize this overall framework to accomplish these goals. And I want to just kind of cover them because I'm sure you've seen a lot of them in the literature, and I want to just kind of introduce you to the differences and how we kind of think about these differences. In general, they all sort of involve 4 major steps. The first is to index a 2-dimensional surface through a variety of different techniques. And that is really the place where the most kind of creativity and differentiation has occurred amongst these techniques. Then the tissue is applied on top of this 2-dimensional surface. And the RNA is sort of transferred onto the surface in a variety of techniques to the actual primers, the DNA primers that are present in this spotted array, or in this surface. And then downstream of that, the methods all have a lot in common. I'll tell you a little bit about the molecular biology, but I won't go too much into detail because they're pretty straightforward kind of molecular reactions to get the RNA, once captured on the surface, to a next generation sequencing compatible library format. And then, of course, the biggest challenge of all is what to do with the data processing. Faye started to cover that, and I know it'll be covered later. So again, from an experimental perspective, the ST methods are most distinguished by the barcoding strategy and surface used. So the original strategy, and one that's been adopted by a couple other groups as well, is to utilize microarray-based spotting of barcoded primers onto a silicon or other kind of surface. So instead of a microarray, just you put down a soluble slurry of different compounds or molecules. In this case, you are barcoding the surface so that you can put down a tissue and capture the RNA onto the tissue. This was the first demonstration of these tools I mentioned already, spatial transcriptomics, which has now been adapted to be sold by 10x Genomics in the platform called 10x Visium. The technology now is at the 50 micron resolution, but it is reportedly going to get better in future iterations of the 10x platform. There's another family of techniques that utilize beads as a way of barcoding a surface. And this is our technology, especially SlideSeq, which utilize 10 micron beads. Each bead has a unique barcode. And then we fabricate the array to have a very dense coverage of these beads along a 2-dimensional surface. And we index the locations of each one of those barcodes on those beads using a very simple in situ sequencing strategy, again, so we can bank those indexed arrays for downstream experiments. And the nice thing about beads is that they can be nicely packed together. They aren't liquid, so you don't have to worry about, when you spot 2 liquids next to each other, they obviously can run into each other. So you can get very, very nice packing of the surface in that way. And we can get down to, to sort of like small numbers of microns with existing DNA synthesis tools and existing beads that we can do that are compatible with synthesis. There's now another family of strategies that utilize polonies or nanoballs as a way of indexing a surface. So individual molecules are spotted down onto a surface that have a unique randomer on them. And then that randomer is amplified locally, either through rolling circle amplification or through bridge amplification on an Illumina sequencer. And then that spot then can be indexed through in situ or through sequencing of that slide to get the locations of those barcodes. And there, StereoSeq is the strategy that utilizes the nanoballs, and PixelSeq and SeqScope utilize the Polony strategy. And then finally, DividSeq sort of does things a little bit in reverse. It utilizes a microfluidic system to cross two different orthogonal barcodes in a 2-dimensional pixel array. And then those combinations of barcodes are then actually reversely embedded into the tissue. So they go into the tissue and capture the RNA within the tissue. And these strategies cover some range between 10 and 100 micron resolution. So the next step is the capture of the RNA onto the tissue. And this process is basically just a couple different strategies. One is to use protease permeabilization, so gentle protease release of the RNAs so that they can diffuse to the surface. Slide-seq utilizes a slightly different strategy where we just take fresh tissue and we slice it directly onto the array, and the freeze thaw of the tissue actually melts the tissue and pushes the RNA onto the surface. And then I guess I already mentioned it, but there are a couple of technologies, DIBIT-seq, which I said already, but also the nanostring platform that basically embed the primers into the tissue by creating a soluble liquid barcoding surface that then can be released into the tissue. I won't cover this for more than a few seconds, but the way that the libraries are generated once the RNA is captured and reverse transcribed follows just kind of like wonderful technology that was developed more than 10 years ago and hugely exploited in single cell analysis. So the two major ways this is done are through template switch amplification, where template switch oligo is used to kind of capture the whole RNA. There's also been additional kind of adaptations of this that and improvements where second strand synthesis utilizes a randomer that is tagged, and it comes in and it primes off of the first strand and gives you additional transcripts that are not full length, but gives you more coverage of the transcriptome. And the other sort of family of methods, which aren't quite used as much because it's a little bit more laborious, although the process is linear in amplification as opposed to exponential, is the use of T7 amplification, the framework that was developed for single cell analysis called CellSeq. So now you've done your library prep and you've done your sequencing. I just wanted to briefly cover some of the important QC metrics that we see as really essential for, for really high quality benchmarking of these technologies, and also assessment of what the capabilities of the technologies are with respect to each other. And the reason to sort of emphasize this is that tissues vary dramatically in their RNA content, the density of the RNA content, their distribution. So some areas of tissue are highly dense and others are not. And so, sort of absolute quantifications of capture efficiency really rely on a faithful segmentation of the tissue into a consistent feature in order to really be able to make comparisons both across conditions, if you're doing different technology development, or across different technologies. So what we like to sort of do is to segment a particular feature. And we're looking here at the hippocampus just because it's so easy to see these features. And we look at the expression of individual genes within this feature. And since the feature has now been segmented, we can now easily compare across different methods as a function of the area that we've segmented out. And then for the resolution, which is the other major parameter of these technologies, we can look at this individual feature and quantify for individual genes, or for an aggregate of all genes, just number of transcripts. We can quantify the thickness of this feature as a way of ascertaining whether diffusion has kind of occurred across this feature. So here we're giving, as an sort of toy example, a few different methodologies, and you can see that the crispness of this profile as we go across this axis changes, and that would be what would be expected as diffusion gets worse or better. And this would tell you kind of what the rough resolution is of a particular technology. This particular strategy is not used universally in the field, but we really hope that strategies like it are kind of more consistently used so that we can do a better job of really understanding the limitations of the resolution and capture efficiency of these technologies. So again, I mentioned the important parameters here. The tissue type is absolutely important. Also, the fixation process that's used can dramatically affect things. And we also have to be cognizant of sequencing efficiency and depth. I'm going to skip over this because I've already basically mentioned this. This was just the example from our paper of this actual analysis. And then I'll just conclude, because I know Faye has also mentioned this already. A key question is how to deconvolute these data. And so we're really fortunate that there are a lot of really great techniques out there that allow us to look at the quantification of the mapping of individual single cell profiles onto these cell types. And now there's also emerging methods that allow us to look at gradients of expression across these features as well. So I think for the future of SST, there's a lot of really great biological applications that Faye mentioned already, developmental trajectories. I think applications to look at pathology and tissue pathology is really exciting. And also just the construction of large-scale atlases of tissue. And I think there's also really great avenues for further technology development. Because it's sequencing based, it can be easily integrated with perturbations that are themselves barcoded like guides. I think there are ways that we can utilize technologies like this to quantify molecular proximity with respect to individual biomolecules. And then also this multimodal aspect that I've already covered as a major advantage of SST. So with that, I'll turn it over to Stephen. I know I'm a little bit over time.
Great. Thanks, Evan. That was an amazing talk. I think we'll proceed to Stephen's talk and then do questions together at the panel discussion. So I'm excited to introduce Stephen Wang, an assistant professor in the Department of Genetics and the Department of Cell Biology at the Yale School of Medicine. Stephen did his PhD at Princeton University in molecular biology and as a postdoc at Harvard in Xiaowei Wang's lab, co-developed MERFISH, a single molecule multiplexed imaging technology. His lab now develops and applies state-of-the-art imaging-based omics approaches to understand the spatial organization of the mammalian genome and how it impacts cellular states. I'm eager to hear his talk today on image-based spatial transcriptomic technologies, and take it away, Stephen.
Thank you, Sophia, for the introduction. And I'm very glad to be able to share this talk here. Let me share my screen first. All right, cool. Hopefully everybody sees this. So the title of my talk today is a practical guide to representative image-based spatial transcriptomics technologies. And I first want to emphasize, or maybe re-emphasize, that this talk is meant to be a practical guide to best help people who never worked on this to adapt technologies in this field. It's not meant to be a thorough or balanced review. And in this field, there are mainly two categories of technologies. One is multiplexed fluorescence in situ hybridization, or multiplex FISH. And in this category, we'll mainly focus on the multiplex error robust FISH, or MERFISH, as a representative technology. And in the second category, I'm going to talk about in situ sequencing with a focus on a couple of latest developments. So first thing first, multiplex FISH. So all multiplex FISH technologies, they are based upon single molecule fluorescence in situ hybridization, or single molecule FISH. In single molecule FISH, what we have are these oligonucleotide probes that carries fluorescent dyes. They have the complementary sequences to their RNA target. So upon hybridization due to base pairing, these probes, they can tile onto your RNAs of interest, and usually you need a tile of tens of probes so that the fluorescence from the individual RNA molecule stands above the fluorescent background, and each RNA molecule would appear as a fluorescent spot. And using this technology, one can count how many RNA copy numbers there are in each cell with very high detection efficiency. Very powerful technique. But one challenge of a single molecule FISH is conventional single molecule FISH can only image one or a few RNA species of interest at a time. And this is because different RNA species needs to be distinguished with different fluorescent colors and imaged in different fluorescence channels under the microscope. So the challenge here is, how do we efficiently detect and distinguish hundreds to thousands of RNA species, really reaching the transcriptome scale maybe at the single cell, single molecule level in the same single cells. To tackle this challenge, we developed MERFISH, or multiplex error robust FISH, and it's by combining combinatorial barcoding and a sequential decoding strategy that we were able to do this. So in MERFISH, in a first primary hybridization, we actually hybridize a library of these primary probes to all the RNA species of interest. And through this primary hybridization procedure, we actually combinatorially encoded all these RNA species through these various overhand sequences. Now, let's say, for example, if there are 16 different versions of these overhand sequences, and each RNA species is associated with 4 out of the 16 sequences, then each RNA species essentially is encoded with a 16^4 combinatorial barcode. And a 16^4 combinatorial barcode gives you hundreds to thousands of different varieties that you can use to encode different RNA species. And now the task is how do we read out these barcodes on the overhangs? That is shown as sequential cyclic kind of a readout fish procedure. So in the first round of this readout secondary hybridization, we have one kind of oligonucleotide probe labeled with fluorescent dye hybridized to the readout sequence number 1. And all the RNA species that have these readout sequence will show up in your imaging as fluorescent spot, and you can mark them with a digit 1. And those RNA molecules that doesn't show up, you can mark it with a digit 0. And then upon photo bleaching or other ways to remove the signal can be removed, and one can apply another secondary probe that read out the second digit in this barcode. And this procedure can be performed over and over again until all the digits in this barcode are read out. And by design, these different barcodes is associated with different RNA species, so we can distinguish the species identity of all the observed RNA molecules. In a real image, it looks like this. So this is the one cell, and different dots are individual RNA molecules. This different colors you're seeing here is actually pseudo color showing the different RNA species after the decoding. And in fact, the whole experiment here is performed with a single color channel for RNA fish. So in this area, there are multiple molecules. Let's focus on the molecule number 2 here. So through the 16 rounds of imaging, we see this molecule number 2 shows up in the 4th round of hybridization, the 7th round of hybridization, the 10th and the 12th round of hybridization. So the barcode that we read out from this molecule is 0001. Note the number 4's hybridization is marked as a digit 1, and then 001, 001, 010000. And then if we go back to our barcode, we know by design, the RNA species that are associated with this barcode is EGFR RNA, and all the individual molecules inside the cell, you can decode it this way. And I do want to mention that besides the combinatorial barcoding and the sequential decoding, there's another innovation in MERFISH that's an error-robust encoding procedure that's borrowed from telecommunication. So in the decoding procedure, we can actually tolerate single-digit errors and correct such errors. With these type of strategies, we were able to efficiently detect and distinguish hundreds of thousands of different RNA species at a single molecule level in the same single cells. So we just heard a great talk about sequencing-based spatial transcriptomics. What are some of the complementary maybe advantages of the image-based spatial transcriptomics, particularly for multiplex fish? One of them is actually the multiplex fish, such as MERFISH, has very high detection efficiency. And people routinely get above 80%, or this is what Evan calls sensitivity, I would say, of detection efficiency. Sometimes it's nearly 100%. So this technology is very good for example, studying low abundance RNAs in single cells. And it's important to point out this high detection efficiency seems to hold true over multiple orders of magnitude of different abundances. So for the RNA species that appears, on average 100 copies per cell, you will be able to detect 94 copies per cell. And for the RNA species that on average appears only 1 copy per cell, in 94% of the cases, you can see 1 copy per cell. It's a very high detection efficiency overall. And then it also has very high spatial resolution, right? So this technique, I think Faye mentioned about this, is not just cell-cell spatial resolution. Actually, you have the nanometer scale kind of a single molecule spatial resolution. And because of that, we actually have 2 levels of RNA information. One is this cell-cell arrangement in tissue, and then also the subcellular RNA localization in cells. And here are some representative works showing, the cell-cell arrangement in the brain tissue or fetal liver tissue. In fact, in this work, we also looked into ligand and receptor signaling gene pairs, how they are expressed and underlying this spatial arrangement. And this is the molecular interaction underlying the cellular interaction that Faye talked about. And then these are the representative works that really studies the subcellular localization of RNA, for example, how RNA are localized in different cell compartments inside of neurons, or RNAs associated with focal adhesions in the epithelial cells. And another advantage of multiplex fish is the ease to combine with our other modes of imaging. For example, in this work, we performed RNA morphish together with highly multiplexed DNA fish technology called chromatin tracing that we can use to trace the spatial folding of chromatin, and also imaging of protein with co-immunofluorescence, and also this WGA staining that actually labels a cell membrane, because this WGA, it can bind to particular glycolipid and glycoprotein on the cell membrane. So you can see that DNA, RNA, protein, lipid, they can all be imaged together in the same single cells. And this multimodal omics or multi-omics capacity is another advantage of multiplex FISH. So let's say you are writing an R1 grant, and in the section where you want to talk about why your proposal is technologically and conceptually innovative, maybe based on the context of your scientific question, you can talk about not only just the benefit of having single cell transcriptome profile together with the spatial information, but also the fact that multiplex FISH have high detection efficiency, have high spatial resolution, 2 levels of RNA spatial information, and this multimodal capacity in that section, right? And then later in another section, you want to write about what are the alternative potential pitfalls of this proposal and alternative strategies. And in other words, what are some of the common challenges for multiplex FISH, and what are the current solutions? So challenge number 1, I think this is a sharp contrast between the image-based spatial transcriptomics versus the sequencing-based techniques. So in multiplex FISH, we need to select a list of target RNA species, right? So it's hundreds to thousands of targets, but it's not the 20,000 unbiased whole transcriptome, right? You still need to pick a list of target RNA species or target genes. So the solution to this, maybe this shouldn't be the alternative strategy, this should be the main strategy, is to use the single cell RNA-seq data to guide your target selection, or maybe use the spatial transcriptome based on sequencing data to guide your target selection. And some common target genes of choice include, for example, cell type marker genes. You can pick this out from the RNA-seq data. Cell state marker genes, you may want to include the cell cycle marker genes. And also for your particular project of interest, you may want to include the project-specific genes of interest based on your domain knowledge. And when people are picking the marker genes, one common question is, how many genes are needed? I mean, how many genes do I need to include in this target list? So I would say, if your goal is to identify these different cell types and cell states with the spatial information, then one question you should ask is, can you still cluster the single cell RNA-seq data and identify the cell types of interest using the marker genes alone? All right, so with the single cell RNA-seq data, if you include all the genes from the seq, you will be able to do the t-SNE, the UMAP, the clustering, and you see these different clusters of cell types using the full data set, right? If you remove all the other genes and just retain your marker genes that you selected, can you still identify those clusters and do this t-SNE UMAP plot and see those cell types using the single cell RNA-seq data? If so, because the multiplex FISH has very high detection efficiency, higher than single cell RNA-seq, it's actually quite guaranteed you will be able to identify the same cell types with the spatial data too. I would suggest maybe start with maybe 5 marker genes per cell type, and you try this analysis. And if that's not enough, try to include more marker genes per cell type. And then usually a few hundred genes are all you need in a project in your MERFISH target list. I do want to mention the maximum transcriptome coverage achieved by multiplex FISH is 10,000 genes, so maybe half of the transcriptome from multiple labs now. I mean, either by combining MERFISH with expansion microscopy, or this super resolved seq-FISH plus. But this, of course, has added level of complexity. So as a start, maybe start with the hundreds of gene kind of scale is a good choice. And challenge number 2, what if my RNAs of interest are highly abundant and or colocalized with each other, like in an RNA granule, for example? So when that happens, the Murphy decoding procedure actually it's likely not going to work, because the different RNA species with different barcode, they will overlap in space, and you cannot robustly decode the different RNA species anymore. And to tackle this, the solution is to use a non-combinatorial sequential single molecule FISH procedure. So image one target at a time. And by design, you can just let one RNA species show up in each round of the imaging. So 3 rounds of imaging will be able to identify 3 RNA species like this, and 20 rounds will be able to identify 20 RNA species. And this strategy can be combined with a combinatorial Murphy procedure. So if you have many genes, most of them are good, they're not highly abundant, and they're not supposed to colocalize with each other. Only some of them are problematic. Then you can actually do this Murphy procedure together with the non-combinatorial sequential FISH as demonstrated in this work. Got the benefit of both worlds. Challenge number 3, RNAs of interest are too short. So like the single molecule RNA FISH, the multiplex FISH also needs to tile each target with tens of probes. We usually recommend 48 probes per target, and that translates to roughly 1.5 kb as a minimum RNA length. So what if your RNA interest is shorter than 1.5 kb? Solution is branched amplification Murphy, as demonstrated in this work. You can use these oligo branches to enhance the signal from each primary probe binding, and RNAs as short as 480 nucleotide could be targeted in this work, and that's with 16 primary probes per transcript. Finally, what about tissue background? What if the tissue background is too high? You cannot see your RNA molecule, and the solution is tissue clearing or split FISH. In tissue clearing, the whole cell is embedded in a gel with the RNA anchored to the gel, and the background causing lipid and protein can be digested away, and RNA molecules can show up when it didn't show up in the unclear tissue. And then in split FISH, people can use these pairs of primary probes to bring down the dilabeled readout probes, and this increases the labeling specificity and leads to a higher signal to background ratio with this strategy. All right, so we talked about how it works, what's the benefit, what's the pitfalls and solutions. How do I start? Well, there are 3 options. The first one is to adapt the technology in-house, and this is doable. The key is you need to build an automated microscope and multi-channel flow system to handle the long and repetitive FISH procedures at least overnight. And no student would want to stay in the micro room overnight, I imagine, to do this manually. So the whole system design is published, and people have adapted this in-house. So this is like a possibility for sure. And then one can find a collaborator maybe with the technology, or work with a company. There are multiple companies with multiple offerings of multiplex FISH right now, and I'm going to talk about where to find them. And we'll start, the first question people often ask is, will the technology work on my sample, right? So I have this particular tissue type and particular tissue preservation method. Does multiplex FISH work with this? I would say check if there were previous demonstration of a single molecule RNA FISH with your sample. But if so, likely the multiplex FISH will work too. If not, that is actually a red flag, and maybe people have tried and failed. And if the single molecule RNA FISH doesn't work in your sample, it's very unlikely the highly multiplexed single molecule RNA FISH will work in the same kind of sample. So unless your collaborator or the company you work with has established the multiplex FISH protocol for your sample, I would say try to first demonstrate conventional single molecule FISH with your sample. All right, so you have your sample, you even have a FISH protocol, and there we have the published protocol, for example. What you need is a probe set that target your RNAs of interest to try first. This is a conventional single molecule FISH. For the probe design, I want to bring your attention to this resource. It's a software package called Probe Dealer on our website, and it can design not only Murphy, but also the conventional single molecule FISH probes, as well as other types of multiplex FISH. And particularly in this section, we talked about how to use this software to design probes just for single molecule RNA FISH as a start. And there's a detailed instruction here. And this is a package we really can vouch for, because this is exactly how we do our own probe design. All right, now let's talk about in situ sequencing. And in the in situ sequencing category, there are two subcategories: targeted versus untargeted in situ sequencing. In targeted in situ sequencing, what you have are these RNA molecules, and they are first reverse transcribed into cDNA. And then these padlock probes are hybridized to the cDNA, and they are circularized with a ligation reaction with or without a gap-filling polymerization reaction. And finally, this circular probe is amplified through a rolling circle amplification procedure. And finally, either the gap-filled kind of a sequence or a barcode sequence on the circle is sequenced through sequencing by ligation. And then in the untargeted FISC procedure, the key difference is there's no padlock probe, but the cDNA itself is circularized and amplified through rolling circle amplification and sequenced in situ. Between these two technologies, the targeted in situ sequencing has higher detection efficiency. The untargeted in situ sequencing has a promise of transcriptome coverage. So there's definitely a trade-off between the two. Overall, in comparison to multiplex FISH, because of the in situ sequencing procedure involves multiple enzymatic procedures in situ, and some of them can be low efficiency, the overall efficiency is lower than multiplex FISH usually. But people have tried to tackle this challenge and improve the detection efficiency of in situ sequencing through multiple ways. One of them is to combine expansion microscopy with, for example, the untargeted in situ sequencing. And the expansion of the sample make each RNA more accessible to all the enzymatic reactions. And through this, people were able to reach a detection efficiency of 0.37%. This may still sounds very low, but it's actually much better than the untargeted in situ sequencing before. And this has the whole transcriptome coverage, right? That's definitely a very significant benefit of the untargeted in situ sequencing. And in this work, thousands of genes, including splice variants, could be detected. And then in targeted expansion microscopy, people did another improvement, which is to use this splint R ligase. So this is a special ligase that can ligate DNA-DNA junction on a DNA-RNA duplex. So no reverse transcription is needed anymore. You can hybridize this padlock probe directly to your RNA and circularize it and then do this rolling circle amplification. In this work, people achieved same work actually as the last one. Detection efficiency, very impressive, 62%, and 8 or 16 probes per transcript were used. So this is good for the short transcript too. And then about 300 target genes were detected. And then there's another technique touched upon by Fei also, the star map technology. And this also avoid the very low efficient reverse transcription procedure by using a pair of linear and padlock probes. So the padlock probe now can be ligated on a DNA-DNA duplex with normal DNA ligase. And in this type of work, people achieve the detection efficiency comparable to single cell RNA sequencing, so about 10%. And 8 probes per transcript was sufficient, and this is good for short transcript. And about 3,000 target genes were detected in the latest version of StarMap. So finally, where to find collaborators or commercial options? I'd like to have a special shout out to my Yale colleague Fan and the Georgia Tech colleague Macosko. They organized this very good platform called Spatialomics.net with weekly seminars from both academic and industry, all the companies out there. Currently, series number 7 is in progress, and all the talks are on YouTube available publicly. If you check into this, you will find multiple offerings of how to start a trial. Finally, thank you all for being here, and I'd like to answer any questions later.
Amazing, thank you so much, Stephen. I wish I had learned all of that at the beginning of my PhD. That was a great practical guide. So yes, again, if you have any questions, please send them in the Q&A. And we have one question from Nusrat, which is, can you use the spatial transcriptomic approach to human skin graft rejection model? So I'd like to pose that question to you, but more broadly, what does the perfect sample look like for spatial methods? And what's an example of samples that would be lacking in applying these tools?
Who were you posing it to? Sorry.
All right, so to each of you, I think
each of us.
both on
Okay.
the molecular side and also on the sequencing side.
Yeah, I can go ahead. I mean, I think so. I have a few thoughts on the specifics around a skin graft model, but I think the main big question that everybody sort of asks or framing it is whether their particular tissue will be applicable. I think that all these methods have done a lot of hard work to try to make the enzymatics and the overall process compatible with a wide range of tissue types. Some continue to be harder than others, like, especially RNase intensive, high RNase concentration samples, like pancreas, for example, or other GI samples can sometimes be challenging for all of these different tools to deal with. Skin can also just be hard because the release of for SST, the release of the RNA can sometimes be challenging. We've had some success with it. We did some work on melanoma in collaboration with Ben Izar's lab. One thing that I think is sort of interesting about your question, though, which is sort of an interesting distinction amongst the different strategies, is that the graft or sort of transplant kinds of tissues have two different genotypes in them. And so you often want to know whether you're looking at a expression effect that's from the donor or the recipient tissue. And that is something where the SST can be particularly useful because you're getting, primary sequence-based data. So you can basically look using SNPs at whether the gene expression is deriving from the host or the donor.
Great. There's a follow-up question on what method could be better than the others. I guess when you're thinking about doing a spatial experiment, how important are having a single cell reference and all these other parameters as you go into it that you can optimize your preparation to have the best results in your data?
I mean, okay, maybe I can take this, Sophia. I think you asked another question is like, what is the perfect sample in the previous right before that? And I maybe I'll answer that first, is that probably there isn't like perfect samples. And actually different technologies are differently suited to different types of samples. I mean, I think Evan like alluded to that a little bit. An interesting example of that is that spatial capture approaches are not sensitive to like tissue autofluorescence, for example. And so, they may be very robust to the tissues which have lots of autofluorescence versus, I think, there are certain samples which are filled with cells which are very RNase rich or granulocytes that explode and such, and they may be those things may be inhibited upon fixation, and those might be better suited for imaging based approaches, right? And so there are some considerations that are sample specific, and then there's like sample quality. I think probably in terms of sample quality, like in my experience, at least for imaging-based approaches and spatial capture-based approaches, they actually align quite well. Like the better the RNA qualities is in your sample, the better your performance will be. And then there's obviously like, you might want to see if people have performed or tried the approach with your sample before. I think Stephen mentioned that a little bit. It depends on how much. Most technologies work like 80% of the time off the bat on a given new tissue, but you don't want to be in like, if you don't want to go and like spend the time debugging in that 20%, then you can like see if other people have already done that like work for you.
I totally agree. We several of us actually talked about the complementary advantages of the different versions of techniques, right? So I guess which method could be better than others, it depends on what do you want and what is the most important feature to you, or maybe like all of these features are important. You can try both sequence-based and image-based spatial transcriptomics. And regarding the first question, like whether the spatial transcriptomic approach is applicable to the human spinning graft rejection model, I can just quickly answer that. I know there are demonstrations for multipass fish. It's applicable to mouse skin, just native mouse skin for sure. It has been demonstrated. Maybe it's not published yet, but it's doable.
Great, thanks. The next question is for people who are interested in the technology development side. What do you think are the biggest challenges in the field? What would you start working on now if you had the opportunity to be in the lab and hands-on working on something to optimize in either the single molecule side or on the spatial capture side?
Well, I'm most excited about kind of providing additional context for these transcriptional measurements. I think we're very interested in utilizing these techniques in the brain and, just understanding, cell-cell interactions, interactions amongst transcripts with other classes of biomolecules, like, which RNAs are sitting on a ribosome or which RNAs are being trafficked to a particular compartment of tissue. Those are particular measurements that are very important in the brain to sort of understand the function of gene expression and also just more generally of tissue function. So I think the extent to which we can provide additional kind of context and understand interactions amongst molecules, I think that is a really key question for us.
I would just add to Evan's thing. I think, the context could also be in terms of sequence, right? Like, I think something that's probably pretty exciting is like sequences will start changing in the animal, for example, like T cells and B cell receptor sequences, or like somatic mutations, or like in cancer, especially like mutational profiles. Or maybe performing like lineage tracing even on de novo like mutations, either somatic or mitochondrial. And obviously that's like the primary sequence, but then there's like non-sequence driven things like methylation or epigenomics or epitranscriptomics. I think is also another contextual thing that's quite exciting in the future.
Great.
I have a question that I'd like to throw out about. It seems like both of these methods are primarily two-dimensional. With imaging, you're at a given focal plane in the sample, and you've squashed a sample into an array for NGS-based approaches. How much of a limitation is that, or how important would it be to try to make this a three-dimensional technology as well?
Cool. So in the multiplex fish and in situ sequencing world, actually the data is intrinsically 3D. We do have the Z dimension of each of the molecule also, but it's usually in data analysis, people don't really take advantage of the third dimension, especially if the question is about cell level kind of spatial analysis, and people just project everything onto the 2D plane and analyze that way. But there are cases where, for example, maybe if you're studying subcellular localization of an RNA relative to the subcellular compartment of the cell, maybe the 3D information is also meaningful. And when studying thick tissue, in situ sequencing actually have the benefit of being able to perform directly on thick tissue with maybe 10 layers of cells. For example, in star map that was demonstrated. For multiplex fish, usually the tissue section is one layer of cell, but one can actually computationally stitch consecutive tissue sections together and build that Z dimension for the cell arrangement also. I think that's the usual way to tackle that.
Thanks.
And well, I was going to say,
yeah,
I do think it's important. There are some ideas and strategies for reconstructing in the third dimension through alignment of serial sections. I think Evan can actually comment on that because we've been building a large scale atlas of the mouse brain, and he's been doing a lot of that. So maybe you want to.
Yeah, I actually started to talk, but then I realized that my mic was muted. So I think it is an interesting question. Actually, I think in the case of the SST technologies, the real limitation there is just the costs of doing the sequencing, because in theory, it's actually not that hard to scale these experiments up and do many serial sections. It's just we literally just can't pay for the density of sampling to reconstruct an entire tissue. We have done that with funds from a generous NIH project to do 114 serial sections of a single mouse brain. And so these sections are spaced 100 microns apart. And, I think that dimension is really key, especially, well, I'll say, so Faye mentioned that the reconstruction by serial sections, there's a couple of nice initial computational techniques, like Barbara Englehart's lab just had a bioarchive preprint where she used a Gaussian process model to basically impute the expression between sections. I think, those kinds of strategies will work. Of course, they'll work much better if you can just serial more sections in these individual spaces. I would say the last thing about that where it is quite important, I think, to get good sampling is when you start to look at, pathological features. So there's a lot of interest, for example, in looking at, amyloid plaques or other extracellular, histological lesions, let's say in the brain, or, tumor responses. And, it is actually very hard to impute the Z-axis without a lot of, actual data from serial sections. So I think that to some extent it's possible just from a tissue organization framework using the strategy I mentioned from Barbara Englehart's lab. But I think for like pathology or real hypothesis generation from the data, I think you really do need serial sections.
We have a few more minutes. If folks have questions, please enter them in the Q&A. So no one has mentioned FFPE. Is that just completely off the table for spatial transcriptomics methods?
I don't think so. We've been working pretty hard on FFPE because I think it's quite important in terms of sample access. I think one issue is that there's like different quality, like FFPE is a general term that describes a variety of tissues that have been banked for a variety of years in different states, and there's just a lot of heterogeneity in FFPE. So one challenge, and I meant a challenge in like a good way, is like, one, how do we like determine what are the features in the FFPE tissue that actually generalizes to good performance? Because I think it is definitely possible. It's just you have to deal with that variability still too.
Definitely, yeah. So in the image-based spatial transcriptomics world, there are even multiple commercial offerings of protocols on like FFPE sample. But indeed, as Faye said, the key of whether your sample is going to succeed with these protocols and these offerings is actually how good is your RNA inside of those FFPE sample. Do you still have RNAs left? So if it's a sample that's prepared, for example, without consideration of RNase-free kind of handling, and it's stored at room temperature for years already, there's a chance that there's no good RNA left, and no matter which kind of RNA targeting technique is going to yield anything. All
right, thanks. Sophia, are there any other questions you'd like to pose to our panelists?
Yeah, I have one sort of long-term question. It seems like the two different methods are converging. The capture-based methods are trying to improve in their spatial resolution, and the single molecule-based methods are trying to improve their multiplexing. Is there a point where you think that both methods will accomplish the same or very similar things, or will there be different pivot points for the methods in the longer term?
Very good question. I was going to say, indeed, the current goal of the image-based spatial transcriptomics is to try to, for example, retain the high detection efficiency while including more and more targets into the library, maybe, and to try to achieve the transcriptome scale. And I think there's a hope that maybe that's going to be finally achievable. There is a considerable technical hurdles to really push to that step. And once people get to push to that step, and how widely that very version is broadly adaptable is another question. I mean, like maybe the technique would be just too expensive or take too long time for each iteration. So I would say, for me, at least in the next few years, I do see the two areas of technique maybe still offering complementary benefits, and it's not like one is going to totally replace another or vice versa.
I was actually going to say that, yeah, I mean, I can see there being some synergistic overlap wherein they merge. But one thing I was going to say actually is that I think it really matters how easy it is to do the to apply it, and how generalizable or how accessible it is. That's probably at the end of the day what matters. Like, I think most people are not going to care about an extra 5,000 genes or 10,000 genes, but what you actually care about is can I run this assay? How reproducible is it? What's the data like?
So one follow-up to that is, how easy the methodology is to actually apply to a tissue, but also how easy the analysis might be, right? How accessible the analysis
that's a great
is.
Question. And I think that's where the call to arms should be, is that we need more accessible analysis pipelines. And this is not it's not saying that the reason we don't have it is people haven't been developing it. I think the reason why it's hard is that each spatial, it's not the same as a single cell problem where you can run a single analysis for every single cell data set that you got, and you get to a point where, well, maybe that's overgeneralizing, and Evan, you can tell me if that's not true, but it is very boutique. But in the spatial side, the hypothesis that you need to answer require oftentimes boutique analyses, and the question is, can we come up with enough flexible, generalizable, and flexible enough computational frameworks to be able to take any hypotheses that people have and fit it into those frameworks to answer the question. I think that's probably what we need, instead of here's a one size fits all framework that lets you just plug in your spatial data and here's your.
I think that's absolutely right. I mean, I think an enormous blessing on the single cell side has been that the noise model of all these all the techniques have the same noise model, and also there's been a lot of convergence and adoption of just a very small number of implementations of these particular methodologies. So basically, methodologies that have the same, assumptions about the distribution of data are all going to work. And that's why all these single cell packages do a pretty good job of analyzing any data from any technology. In this case, I think we have two different issues. We have one issue, which is that the noise models are very different. The difference is the big picture between between sequencing and imaging are quite distinct, right? There's the sequencing-based strategies have a more kind of single cell-like Poisson dropout model, whereas source of noise, whereas the imaging-based strategies, the noise is considerably more complex, but perhaps more geared towards false positives. But even then, that's a very loose abstraction of the issues. So anyway, the point is that these methods have very distinct differences that make it hard to implement widely these different methods across different single method computational methods across different experimental methodologies. And then the other issue is on the actual like biology front, whereas I think a lot of the initial questions that people have of single cell data are pretty well defined and small in nature. There's a wide range of questions that people have with the spatial data, and so it can be hard to sort of capture that with a single technique.
I think that's a great transition to the next set of speakers, actually. So thank you so much, Stephen, Faye, and Evan, for giving us an insight into the technology, and I'll let Bill moderate the next session.
Yeah, thank you very much, Sophia. Bill Flynn, who's the Associate Director of Single Cell Biology Lab at Jackson Lab, will be introducing the next speakers who are going to talk about some of these areas in terms of analytical strategies for particular applications of spatial transcriptomics. Take it away, Bill.
Great, thanks, Mark. So I'd like to introduce our next speaker, Julia Salzman, an associate professor of biomedical data science, biochemistry, and statistics at Stanford University. Julia received her bachelor's at Princeton and her PhD at Stanford. And her lab now focuses on computational and statistical methods to understand RNA biology regulation, particularly splicing, in single cell and spatial contexts. Hopefully you saw some of her work in Science maybe 2 weeks ago as part of the Tabula Sapiens Consortia, describing the creation of a multi-organ single cell proteomic and transcriptomic reference. And so looking forward to her talk today, unraveling the spatial regulation of RNA isoform expression. Take it away.
Thank you. That is great. That being that it looks like my slides are successfully sharing. Is that true? Okay, great. And thank you for the introduction. I've also really enjoyed all of the talks from the earlier part of the session. And I'm excited to tell you about some of our work today that is what I'll tell you about today is exclusively focused on the analysis side. And I'm going to give you a taste of two projects. They are led by a group of fantastic people in the lab. Rob Bierman, who is a graduate student, has done the vast majority of the work on analyzing hybridization-based approaches like merfish and seqfish. And he is joined by Rusbe, Caitlin, Julia, and Elizabeth, who have each contributed enormously in different ways to his project and a second one that I will talk about. So I'd like to remind myself and everyone that there is a major mystery of the human genome's function that remains today, which is that essentially every single gene in the genome has more than one variant of it that is expressed. So we talk about genes in biology and molecular biology, but actually genes are not single things. They are the genomic or the DNA blueprint that allows several, 2 to sometimes tens of thousands of different sequence variants to be transcribed and produced in a cell. And the reason that I'm especially interested in this is that over the years, molecular biologists have done painstaking, very difficult, and very important work characterizing small numbers of different variants of the same gene, which I'll call isoforms, and found some very fascinating, important functional features of them. They are crucial in development. Their dysregulation is now known to cause myriad diseases. Probably the first disease is known to be caused by dysregulated RNA isoform expression, weird neurodegenerative diseases. But today we appreciate that they also cause diseases like cancer and other complex diseases, autoimmune diseases. And as a snapshot of perhaps why, although I think this is an incomplete reason, we know that the same gene can make different isoforms that produce proteins that actually have different functions, but more than different, sometimes opposite functions. So while there are some genes that have been studied by hand, as I said, that allow me to make these claims about their function and development and disease, the very vast majority, the overwhelming majority of genes in the human genome have isoforms of unknown function. And so we have approached this question using a data-centric view because this is a question that is probably literally impossible for humans to answer using traditional molecular approaches. There's simply too many genes, too many isoforms to imagine that it's possible to study each isoform's function one by one. So what we're interested in is tissue-specific regulation, cell type-specific regulation. And one of the reasons that we are especially interested in spatial transcriptomics is because spatial regulation of isoform expression is an incredibly important potential hypothesis generator for functions that isoforms may play. And in order to make these kinds of inferences, we need robust algorithms that, as the previous speakers really clearly described, we need ones that are robust across technical, biological, and even platform type technologies. So I want to give a quick overview of what the type of isoform that I am going to talk about today. There are two kinds. One is one that I described earlier, which is splice isoforms, and I think most people are familiar with that. Another much more unexplored way that RNAs are regulated at the isoform level is by alternative use of 3' UTRs. And this is, I would say, a very understudied problem, partly because the computational statistical tools needed to define 3' UTRs are actually more difficult to develop than ones for splice isoform detection. Okay, so here's just a tantalizing example. Again, this is a kind of hand-picked example based on beautiful work from Christine Meyer's lab on CD47. CD47 is a cell surface marker, and it can protect against phagocytosis. And it has very remarkable regulation of its 3' UTR. This is not, well, it does have protein coding changes, but the function for the UTR is independent of that. So there are two different variants at the 3' end of this transcript. One is the short version, and the short version, when it's expressed, the protein localizes to the endoplasmic reticulum, leading to increased phagocytosis. The same coding region, however, when expressed proximate to the long UTR, produces a protein that is localized to the plasma membrane, and it protects against this phagocytosis. So it leads, just the non-coding region of this gene leads to very different functions for the gene. And so, this is one example. And our interest is in trying to identify much more systematic, unbiased ways of assessing functions for the UTR. And so I want to tell you about how we are approaching this problem. And I'm going to get to spatial, the relationship to spatial transcriptomics in a second. But at first, I want to tell you about the regulation of 3' UTR that is detectable by 10x sequencing, because as Evan and others described, many spatial transcriptomic-based methods are analogous to, or their capture approach is analogous to 10x capture. And so very similar methods can be applied, and I'll get to that later in the talk. Okay, so how could we potentially identify these two different 3' UTRs, for example, in CD47, but more generally? So let's say cell 1 has a short UTR and cell 2 has a long one. If we prime from the 3' end, we expect to see a read buildup in cell 1 near to the 3' UTR that is shorter versus one that is longer, closer, the read buildup would be closer to the longer 3' UTR in a cell type that has it being expressed, okay? And so if you look at the read buildup, the histogram of the read buildups in these two contexts, you'd see a separation of these peaks. This is a very intuitive idea, and it is the basis of many published algorithms based on peak calling. And I don't have time to talk to you about this, but we have papers on this topic that while it's an intuitive and appealing and simple kind of algorithm to engineer, it actually misses important regulation. So we have developed a different approach to detecting regulation of the 3' UTR, which we call the READ-CS, and it's a paper on the bioRxiv. This approach is unbiased in the sense that it does not rely on gene annotation or annotation of the 3' UTR, and it is able to assign every single cell in every single window that tiles the genome a single statistical score. And that is very important for actually for spatial transcriptomic applications, as well as for single cell ones. Okay. I was going backwards. I was like, why is that happening? Okay, so what I'm going to tell you about now is two vignettes. One is how we're using this kind of measurement of the 3' UTR from single cell sequencing to basically make inferences about how UTRs may be functioning in single cells, similar to CD47. And the second one, I will hopefully have a chance to talk about, touch on new methods for how we are applying this approach, as well as an analogous one that we've developed for splicing to platforms like SlideSeq and Visium. Okay, so as I mentioned, spatial, and others have mentioned, spatial transcriptomics based on hybridization and microscopy have complementary powers to fully sequencing-based approaches like a 10x experiment. So 10x, of course, is able to measure the nucleotide composition of every single RNA in the cell. It has high resolution in that realm, but no spatial information, whereas spatial approaches like MERFISH have subcellular spatial resolution capacity, but no sequence level or little sequence level information. So what we do is a data integration in the work that I'll tell you about here. It is that we match on cell types called by each of the two platforms and then do something like a correlative analysis. So here's an example. We'll take a single cell RNA-seq data set, perform unbiased analysis of its 3' UTR. For example, here I'm showing you a mouse brain and two cell types where the algorithm calls very significant differences in the 3' UTR between the two cell types. And then we apply another kind of score called subcellular localization RNA ranking platform, SLRP, which provides us a single cell, basically a measure of how peripheral this gene is in every single cell type compared to all others. And we're going to summarize that by a median SLRP score per gene per cell type. The intuition here, we actually have three SLRP measures, one that is peripheral, one measuring how radially concentrated a transcript is, and one how punctate the RNAs are. And so the intuition here, if you can see the dots, is that the red dots are supposed to represent the particular gene under study. And in this case, it's highly peripheral compared to kind of random, or central here and random in the other plots. And so this gene would receive a low score if it's highly central and a high score if it's highly peripheral. We have a second metric that measures how radial the gene is, and then a third one that measures how punctate the gene is. And I'm not really going to talk more about that here. Because I realize that I'm running out of time, I want to tell you very quickly that we have assessed how technically and biologically reproducible this metric that measures subcellular localization is, and we find high high concordance between biological replicates in all three of the metrics. And then finally, just a last vignette, that the gene that I talked to you about earlier, TIM3, where we found remarkable differences in the 3' UTR use per cell type, we have also found very reproducible peripherality of specific UTR use in this gene. So the short UTR that we measured in an unbiased way through the use of the first statistical metric, reads-yes, correlates very significantly with peripheral localization of the UTR, the mRNA. Okay, so just to refresh you, we're seeing very peripheral localization of these shorter UTRs, whereas much less peripheral localization of the others. And another interesting feature of this plot is you can see there's only exactly one UTR that is annotated in the genome browser. And this highlights what I mentioned earlier, and I think the scope for future investigation, which is that many of the UTRs that are in use in real tissues have been unannotated to date. And okay, so just to summarize, this is an approach that is general, reproducible, and we predict can inform functional hypothesis generation. Okay, in the last 3 minutes, I'm going to try to tell you about the another approach that we have in preparation, which is the application of statistical measurement of both splicing and UTR use to spatial transcriptomics. Okay, and I'm going to tell you about an example with this in the splicing z-score, using the splicing z-score, which is going to replace, intuitively, it will replace something like gene expression. So typically, spatial transcriptomics is reducing all gene measurements to gene expression, like summed over all possible RNA isoforms. Here, we are going to replace that measure with a measure of, for example, how small on average the introns are, which we call the splice, and it can capture and does alternative splicing differences between cell types. You can read this paper, this work in a paper that I've linked to. Okay, so last slide here. I want to give you one of many examples that we are investigating, which is an example. This is Visium data made public by the company in kidney and brain. And here I'm showing you the histology, the gene expression of this gene MYL6, which is an actin light chain gene, which is ubiquitously expressed. So it is thought to be kind of a housekeeping gene. One of the fascinating outcomes of the Tabula Sapiens project, and one that I imagine is going to only become more prominent as we analyze more data, is that genes that we consider to be housekeeping genes are actually tightly regulated at the cell type level, and we believe also likely at the spatial level, which is very important, I think, and interesting for genome science. So one thing that you immediately see here is that gene expression is kind of noisy, not really spatially, regulated, whereas the splicing of this gene is very, very distinctly regulated, with much more exon skipping in these upper and lower fragments of the tissue. And we can formalize this difference between gene expression and splicing by using an analysis, residual analysis, regression analysis, and other analysis that I don't have time to tell you about. There is no correlation between gene expression or read depth and the splice. Okay, another example is in a ribosomal protein gene, RPS24, where the regulation in the spatial realm happens through fascinating regulation of inclusion of a microexon that is only 3 nucleotides long. Okay, so I didn't want to stop in time and thank all of the team members. Some of them were not able to get their pictures in time, but thank you.
Great, thank you, Julia. So I think in the interest of time, I think we're going to go into our next speaker and we'll have questions at the end. So it's my pleasure to introduce, I think, our last speaker of the day, Lyla Atta, who's an MD PhD student at the Johns Hopkins University, currently pursuing her PhD in the Gene Fan Lab. She uses mathematical modeling and computational methods to elucidate complexity and dynamics at single cell resolution. Her recent work has included developing reference-free cell type deconvolution methods and improvements in how we embed, visualize, and analyze inferred RNA velocity kinetics. And so today, she's here to describe developments in spatially contextualized changes in cell types, or sorry, cell states. So Lyla, please take it away.
Great, thank you so much, Bill. Hi, everyone. My name is Lyla Atta. I'm an MD PhD candidate, currently working in the JeffWorks lab at Johns Hopkins. And today I'll be talking to you about how we can leverage transcriptomics data to understand the dynamics of transcriptional cell state changes and how we can extend that to spatial transcriptomics data to begin to integrate our understanding of the spatial context of tissues when probing cell state changes. And towards the end, I'll talk a little bit about the current analytical challenges and opportunities that we're working to address to achieve this goal. And so before I begin, I'd like to thank you all for being here and thank you to the organizers for putting on a great event. And a big thanks to my advisor, Gene Fan, and my lab, the JeffWorks lab, for being a wonderful source of support and guidance as I explore the questions I'm about to talk to you about today. So first, I'd like to begin with a bit of background as to how I got interested in this question in the first place. So by the time I was finishing up my undergraduate degree, I had been involved in several projects that were looking at cells' kind of short-term response to drug treatment in a variety of different tumor contexts. And so for this reason, I was interested in understanding the dynamics of cells' transcriptional state changes at a single cell level, kind of in the hopes of better understanding the mechanisms of response and resistance to drug treatment. Around that time was when all of the high throughput single cell transcriptomics methods were really coming online in an accessible and reliable way. And this was great because it was a tool that allowed us to make these system-wide whole transcriptome measurements at a very high cell resolution. And as we've discussed already, these measurements allow you to take complex tissues composed of cells of different types that are doing different things, measure the amount of RNA of every gene in every cell, which will eventually give you an insight into what each cell is doing and what kind of functions it might be performing. It was, however, a little bit unfortunate that all of these high throughput transcriptomics measurements destroyed cells to capture RNA, because that meant that they could really by design only provide snapshot measurements. And so there are both experimental as well as analytical ways to try to get around that. And one of the analytical approaches, pseudotime inference, relies on ordering cells based on the similarity of their transcriptional profiles. And this allows you to infer what sequence of cell states a cell might go through over the course of some dynamic process. And so this is kind of like looking at a picture of people jumping into the water. Even if you didn't know anything about jumping or about gravity, you can kind of intuit that these two people up here are in similar stages of their jump trajectory and are in a very different stage than this person down here. However, with this pseudotime approach, you really aren't able to follow what an individual cell is doing over time. But luckily, by the time I was starting my PhD, a new approach called RNA velocity had been around for a couple of years that went one step further from these pseudotime approaches to begin to actually infer what a specific cell might be going to do sometime into the future. And so RNA velocity works by looking at the relative amounts of spliced and unspliced forms of a gene to infer that gene's gene expression dynamics. So in gene expression, a gene starts getting transcribed to create the unspliced pre-mRNA that has introns in it. That then goes through splicing to remove the introns, and the spliced mature form of the RNA will eventually get degraded. And so when a gene first gets turned on, you'll expect to see a lot more unspliced RNA since splicing and degradation haven't had time to catch up yet. And then given time, gene expression will reach some steady state where there will be a stable ratio of unspliced to spliced RNA. And then when a gene is turned off, you will see more spliced RNA than expected from the steady state ratio because the unspliced RNA have stopped being produced and the spliced RNA have yet to be degraded. And so you can do this for, if you plot the spliced and unspliced RNA for a single gene, you might see something like this. Each point is a single cell. The black line indicates the steady state ratio of spliced to unspliced RNA. And if a cell is above the line, it has more unspliced forms of this gene than what you would expect at steady state. And you can infer that that gene in that cell is being upregulated. And then conversely, if a cell is below the line, it has more spliced RNAs than expected at steady state, and you can infer that that cell is downregulating this gene. You can do this for every gene in a cell, and you'll get an overall RNA velocity vector for that cell that tells you what that cell's transcriptional state is predicted to be sometime into the future. And so for real data, it looks something like this. Here I'm showing a PCA plot of single cell data of neuronal development. Each point is a single cell, and the arrows represent the average RNA velocity vectors in that area of the plot. And so you can see that the arrows follow the trajectory you would expect from known biology, from radial glia to neuroblasts to immature neurons, and then finally to mature neurons. And so there are a few different things that you can do with this RNA velocity analysis, and this includes determining which genes are exhibiting significantly dynamic behavior, which genes have differential dynamics between cell types. But to kind of get an overall sense of the dominant trajectories in a single cell population, we still really rely on looking at the population in reduced dimensions and projecting the RNA velocity vectors onto this two-dimensional representation, as I'm showing here with this neuron development data. And so commonly used dimensional reduction methods that can be used to generate these two-dimensional representations take into account the gene expressions in your single cell data, but don't take into account the RNA velocity information. And so we hypothesized that taking into account RNA velocity would give us a more reliable representation of the single cell trajectories within single cell transcriptomics data. And so to test this hypothesis, we developed VeloViz, a method to make low-dimensional representations of single-cell transcriptomic data that takes into account predicted future transcriptional states inferred from RNA velocity. And we found that it can indeed yield more reliable representations of single-cell trajectories, especially in cases when the collected data is missing some intermediate cell state. And so as we begin to consider all of the contextual factors that come into play in influencing cells' transcriptional state changes, one of the important ones that that has been the main focus today is cells' interactions with other cells in their immediate spatial vicinity. And so to get a better sense of how cell spatial context influences their transcriptional states, we can adapt this RNA velocity model to use information we can measure from imaging-based spatial transcriptomics methods like MERFISH. And so here, instead of modeling RNA splicing dynamics to infer expression dynamics, we model RNA nuclear export. And so here, cytoplasmic and nuclear RNA are analogous to the spliced and unspliced RNA in our previous model. And so in a similar way, if you have more nuclear RNA for a specific gene than what you might expect at steady state, you can infer that a gene is being upregulated. And then if you have more cytoplasmic RNA than what you would expect, you can infer that that gene is being downregulated. And so Xia et al. Applied this model to MERFISH data collected from an osteosarcoma cell line in culture and used it to infer RNA velocity. And so here is an image. I think you might have seen this in one of the prior talks that they collected, and each one of these spots is showing one single RNA molecule. And below here, each of the spots is pseudo-colored according to what gene that RNA spot corresponds to. And so because this data was collected from cells in culture, we expect that the dominant cell trajectory in this data to be progression through the cell cycle. And indeed, when we visualize the transcriptional states and RNA velocity in an RNA velocity-informed embedding, using VeloViz, we see a very clear cyclic trajectory. However, cells and culture is sort of the kind of ideal case scenario to apply the spatial RNA velocity approach to, because cells are fairly well separated. They're all positioned in similar z-planes. And so fairly standard cell and nuclear segmentation approaches can be applied to determine the nuclear and cytoplasmic RNA counts needed to run RNA velocity analysis. And so extending this approach to cells and tissues will likely prove to be much more complicated, but also a lot more exciting in terms of the biological questions we get to ask. And so here are some of the questions we've been thinking about as we think about applying RNA velocity analysis to MERFISH data in tissues. While we're specifically thinking about these questions in the context of in situ RNA velocity analysis, they're definitely not specific to this analysis approach and are applicable when using imaging-based spatial transcriptomics data to answer biological questions in general. And so one of the first things to consider is that dealing with this data is not a trivial computational task, considering the size of the imaging data. Depending on what type of analysis you're interested in performing, you may not want to abstract the data all the way to a single cell counts matrix and position matrix, which is kind of the most summarized form of the data. So for example, if I'm interested in applying RNA velocity to a particular MERFISH data set, the most abstracted data I can use is a collapsed z-stack of the nuclear and cytoplasmic stains and the RNA spot coordinates. Depending on the architecture of the tissue, I may want even one less level of abstraction than that, and I may want to take into account the variability in cells' vertical positions in the z-axis. All of this is to say that data storage, transfer, and computational resources needed to work with this data become kind of a non-trivial consideration, as we've discussed earlier on, especially for groups that are not used to working with this data type. And so, as I mentioned, RNA velocity analysis relies on looking at the relative abundance of nuclear and cytoplasmic counts. And this will rely on accurate segmentation of the image to identify nuclear and cytoplasmic regions. And as I mentioned before, this isn't an easy task for tissues where cells might be densely packed, may have complex morphological features, like, for example, different projections in neurons. And so traditional segmentation approaches have kind of relied on nuclear and cytoplasmic stains to aid in informing segmentations. But newer, more sophisticated approaches now take advantage of deep learning methods and the additional RNA profile information from the high throughput transcriptomic measurements to better inform segmentation. And then finally, determining the RNA count for cells and subcellular compartments relies on multiple steps. Each has an associated error rate. So in MERFISH, for example, we identify RNA species using a binary barcode, and that is determined from several rounds of sequential FISH. Counting RNAs relies on an accurate reading of this binary barcode and any subsequent error correction. And so each of these steps is associated with some confidence level. And in sequencing data, by comparison, we have a standardized method for reporting confidence in sequence-based calling. But we don't have a method that we've come up with that acts in an analogous way for imaging-based spatial transcriptomics data. And then in the next step, when assigning RNA counts to individual cells, and in the case of RNA velocity to individual nuclei, we need to be able to take into account our confidence in this RNA count assignment, especially if segmentation schemes are more complex or dealing with more complex tissues. And so quantifying our confidence in each of these steps and understanding the extent to which any downstream analysis pipelines, not just RNA velocity analysis, is kind of sensitive to error propagation will be essential to evaluating these analysis methods' robustness. And overall, this will allow us to assess how confident we can be in the biological conclusions we draw from these analysis results. And so with this in mind, we can begin to ask really interesting biological questions, going back to some of the questions I mentioned in the beginning and what we talked about earlier. So for example, how do developing tissues organize themselves into structures, and how do these processes begin to deregulate to give rise to some structural abnormalities that we might see? How do emergent properties arise specifically from the different cell types in a tissue as they interact with one another and perform their functions? And finally, coming back to the question that got me personally interested in this field, how do cells and tissues respond to drug treatment in a coordinated manner, and how might these mechanisms lead to therapeutic resistance? And so with that, I'm happy to discuss more and take any questions.
Great, thank you so much. Just to remind everyone to, if you have any questions, please shoot them over in the Q&A box, and we will try to get them answered. But while we are waiting for questions to trickle in, I just wanted to follow up briefly on what you just mentioned, Lyla, which was, the spatial technologies are really allowing us to start to interrogate kind of multicellular structures, or maybe even extracellular structures like collagen fibrils and things like that. What are your thoughts on where to start, how to start interrogating those kind of multicellular structures? Yeah, any thoughts?
Yeah, I think that comes back to one of the points that we discussed in the earlier panel. I think that it'll hinge on our ability to make these multimodal measurements and how well we'll be able to integrate the different data types from these different modalities.
Can I just ask a clarifying question, Lyla? When you're inferring pre-mRNA versus mRNA from MERFISH data, based on whether the transcript is detected in the nucleus or the cytoplasm, is that right?
Yeah.
Rather than specifically around detection of intron sequences from single cell data.
Yeah, exactly.
Or do you use that at all? Do you use anything do you borrow anything from the single cell data in terms of determining pre-mRNA state?
Not in what I presented, but you can for sure. I think one of the things you can do is integrate single cell RNA sequencing data, which will have the intron exon distinctions in the sequencing information, and in a similar process to what Julia was describing with her work, inform the RNA velocities that are inferred from the single cell data in a spatial context with the MERFISH data.
Okay, great, thanks.
And so I know Julia might have to leave a little bit early, so maybe some questions targeted towards her. One particular question is, rather than looking at a summarized expression, measure of gene expression, when you're looking at isoforms, how limited are you by sequencing depth? And are things like long read sequencing or something like that something that could be applicable to this?
That's a great question. One interesting thing is if we, even if we consider 10x, because it is 3' biased, we can actually get a fair amount of information about the delta 3' UTR use. And that information is actually not limited by, the traditional thinking that 10x is shallow, because really every single read does inform us about the 3' UTR. For splicing, for observing splice junctions, yes, we do have both a bias to observing those exons at the 3' end, and so some limitations about what we can observe directly. But at the same time, because the approaches that I talked about in the first part are based on data integration from kind of orthogonal experiments. Every day there are more and more experiments done and published on the short read archive. So basically our inference, we haven't tried to do this, but we could, I think, bring the sequencing depth really close to like infinity almost, because of how many resources are being devoted to it in cell types or tissues that are well studied, like the mouse brain. The answer would be slightly different in less well-studied tissues and organs.
That kind of touches on another question I had, which is how do you, more generally, and anyone could address this, how do you know you have enough data?
Okay, I'll answer it. My answer is, as a statistician, there is never enough data. There's never too little data, and there's never enough data. The amount of data tells us how noisy our inference is, how reliable it is. So I am a firm believer in one read tells me more, is statistically, information theoretically richer than 0 reads. So I will never impose like very strict filters on, like intuitive filters, like we need 10x coverage or 20x coverage. That's my view.
Yeah, of course, there's no simple answer to that question, I know, but I'm just thinking about in terms of doing quality assessment on a data set, looking at number of barcodes, number of transcripts detected. How do you think about whether or not your experiment worked, whether the experiment worked, but you need deeper sequence? Looking at isoforms, you obviously probably need more depth than looking at 3' UTR, et cetera, et cetera. But I'm curious as to how you approach that kind of setting an a priori expectation when you go in, dial up the NovaSeq and count how many reads you expect to get, and then whether or not that was sufficient to answer the questions you have. You look like you were going to say something.
Oh, me? Well, I think it's a great question. I mean, we are not currently like thinking about how many NovaSeq reads to run. One thing that we do is basically make projections. So there's this really famous historical problem that I'm sure everyone is familiar with about estimating the number of unseen species. And it's very historical, like probably more than a century old, and it has a lot of applications today with basically, to your point, how do you know, given that you've seen certain numbers of barcodes or some number of RNA species, how can you project without actually, how can you best guess how many you would see if you sequence 10 times deeper? And if that number is high, then I would argue, yes, spend the resources to do that 10x more sequencing. If that number is low, then maybe those resources could be better allocated somewhere else. And so what we have done is, and I don't have the numbers off the top of my head, but we basically tried to infer, given that we've sequenced at this depth and we see this number of isoforms or regulated cell types specifically, how many do we think are like over the entire genome are? So that's my answer. I'll let other people give others.
Oh, I was going to say that, sorry, I just want to echo that. I was going to say we often use downsampling to make empirical projections of where we're going to be if we sequenced more, and we use that to guide if it's worthwhile to sequence more.
One follow-up there is, I mean, especially as you're transitioning to spatial, right? All the droplet-based technologies, right, have some bias, right, based on what size cell will fit inside a microfluidics device, or what is sensitive to dissociation, or things like that. And so when you're moving to spatial, now you might be able to profile cells that maybe you don't have high-quality data to understand what they are. So what, any thoughts on what to do there to kind of address, if you're using single cell data to deconvolute your spatial data, but your single cell data is not complete, what do you do?
Oh yeah, this is a great question, because we've been developing a lot of approaches to deconvolve spatial data from single cell data. And obviously there's this like out of reference question, which is like, what happens when you have stuff that's on your spatial data that's not in your reference data set. And I don't know, in general, like, we tested I don't know if there's any method that's actually really great at handling that, and we're working on basically ways to handle the out of reference problem currently in the lab. So that's a great question. Basically, you don't want your thing to, you want some uncertainty. You want it to say that like, oh, I'm not actually quite sure what the cell type is, versus like assigning it to some, you know. And oftentimes what happens currently in our approach is it assigns it to the closest cell type transcriptionally, which you can, you know, imagine is like both like a, you know, you can imagine that's like good and also bad, right? Yeah. Depends on what your tolerance for misassignment is. But that's a good question. I wouldn't say that's a solved problem.
We've also worked on reference-free approaches to kind of decompacting
exactly.
Spatial data. And I think that kind of gets around some of the reference biases that might come into play. Yeah,
exactly. I guess one addition to that is just like, oh, well, maybe also, how do you know, you asked, how do you know if your reference is good? Like, I think it's also important to consider that. And currently, we are not. I mean, people who develop algorithms for assignment assume good references. And I think that's another thing that, I don't know, I guess like we've been working on, is that like, how many cells need to be in a given cluster of your reference, or like, what is the quality of the reference before to assign a cell to the spatial data?
Yeah, I think that's a huge issue, right? Just the quality of publicly available data and their annotations. And so hopefully that's kind of the things that Tabula Sapiens, Tabula Muris, and Allen Brain Institute, and things like that are hoping to solve a little bit. HubMap, for example. So additional questions. So Julia, can you detect various isoforms within the same cell type for a specific gene? Is that something
That's
we have
a great
the resolution
question
to detect?
Oh, sorry. Go ahead.
Nope, that was it.
Oh, okay. Yes. Great question. And actually, that's our one of our major motivations for developing these bona fide single cell resolved measures of splicing and UTR use, because we're preparing a manuscript that basically we want to use these measurements to cluster cells them without using gene expression. That's one. And to the question that you specifically addressed, we want to test if a single cell type can be subclustered on the basis of its isoform expression. And yes, we find pretty strong statistical signals suggesting that cell types have, like a single cell type actually has subpopulations that are splicing genes differently from each other. Yeah.
Great, thanks. And in that same vein, I think the last question in the Q&A for you is, measuring alternative splicing at the 3' UTR is one thing, but what about alternative spliced forms involving exons elsewhere within the gene, not at the 3' end? What methods do you see as amenable to tackle that problem?
Yeah, that's a great question. So, first of all, as poly A priming and 10x, as well as spatial transcriptomics, does have a bias towards the 3' end, but it doesn't exclusively measure transcripts at the 3' end. And like as we sequence deeper, we actually gain enough statistical power in some cases to identify splicing event with 10x or oligo DT priming throughout the gene. That's the first. And then the second is SmartSeq2 is a great way that we can, it's a great technology for measuring splicing across the entire gene body. So we can use that as well we haven't, in the first part of my talk, when I talked about multimodal data integration, we haven't used that, but it is something that would be probably very powerful.
Yeah. Great. All right, thank you. Jeremy asks in the chat, is there any use for bulk RNA-seq data in these analyses? I think that's in the context of maybe the reference free, or if your reference is not particularly great based on single cell data. So I believe the answer is yes. All of these technologies are complementary, right? Bulk RNA-seq, single cell RNA-seq, spatial. But does anyone else have any comments on that?
Well, I guess for the targeted versions of the image-based spatial transcriptomics, definitely, I mean, from the bulk RNA-seq data, one can derive, for example, domain knowledge information about, for example, which genes changes in this developmental process or disease process they are studying. And these definitely worth adding as the target genes in the library design, and one may find, their spatial information, maybe our cell type kind of specificity together with the spatial information. Indeed, so it is important and helpful.
Yeah, that's exactly what I was going to say.
Bill, any other questions remaining?
I don't see any more in the chat. The only other question I have is just, when dealing with trying to understand dynamics, right? And you're taking essentially a single snapshot of a tissue section, or, in a droplet-based world of a tissue dissociation. What are your expectations on whether or not you're sampling all the states necessary to really derive a trajectory?
Yeah, that's a really great question. I think it really depends on the context that you're measuring and kind of what kind of prior domain knowledge you have. We have explored kind of artificially removing some intermediate cell states and kind of trying to understand the extent to which RNA velocity can, through its inference of projected future transcriptional states, can give you some insights into what is happening in this intermediate missing cell states. But at the end of the day, kind of RNA velocity in at least the splicing model is kind of limited by the time scale of splicing dynamics inherently, and so you can really only accurately predict missing cell states or future unobserved cell states to the extent of the timescale of splicing dynamics.
Great, thank you. Those are all the questions I had, Mark, and there's one just in the chat.
All right.
So I think Julia has left, unfortunately, Bruce. So I don't think we can answer your question, but just for the question is, she missed a method that you said would be great to looking at alternative splicing across the gene. I think that she mentioned SMART-seq2, right? So which is a whole transcriptome-based method. But additionally, even 10x, which is poly-A priming, you're going to hit poly-A tracks within introns and exons. And as well, there's some other mechanisms like first strand synthesis hijacking, where in the, trying to capture and build cDNA from an individual molecule, you might get another one at a non-poly-A site. So there are mechanisms within 10x, for example, to get information about introns and non-3' exons.
All right, well, Bill and also Sophia, thank you so much for moderating these sessions. Faye, Evan, Stephen, Julia, and Lyla, thank you very much for these informative and clear presentations. It's wonderful to have you on the session today. Thank you so much for making the time and effort to share your knowledge and perspective with the broader community.